Welcome everyone to our last seminar for this year. I would like to begin by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I extend this acknowledgement to the traditional custodians of the various lands on which you are all joining from today. I pay my respects to their elders past, present, and emerging, and embrace their continued connection to this place. Today we have the pleasure to be joined by Professor Mei Hong from the Department of Chemistry at MIT. And just briefly, Mei has received a number of awards for her creative development and application of solid-state NMR to study the structure and function of membrane proteins involved in infectious diseases. And today she'll be sharing her team's work on the SARS-CoV-2 envelope protein. So whenever you're ready, Mei, you're welcome to begin.
Okay. Thank you, Emily, and thank you, Tracy, for the invitation to your this membrane protein seminar series. And so today I'm going to give you a progress report of my group's recent efforts in using solid-state NMR spectroscopy to study the structure and dynamics of one of the virus ion channels or viroporins, that is the envelope protein or E protein of the SARS-CoV-2 virus. Oops, let me see. Okay, so, oh boy. All right, can you see all these other software things popping up on your side of the screen? Hopefully not, but anyways, okay, so yeah. Okay, so viroporins are a very important class of membrane proteins that have been traditionally underemphasized by structural biologists for lack of, really good and efficient techniques to study those. So these virus membrane proteins conduct ions. They form basically pores through the virus lipid envelope or through the cell membranes in which the viruses are reassembled. And so they conduct ions to cause often pathogenic effects to cells. They usually also have other functions. So these proteins tend to be multifunctional. They can also mediate virus budding and assembly. And so this review article in Nature Review Microbiology highlighted basically the various viroporins known at the time. The prototype of the viroporin family of proteins is the influenza M2 protein, which my group has studied in great detail. But they're really, most envelope viruses have these proteins, and there is enough functional and structural diversity, as we're beginning to realize in our studies of the SARS-CoV-2 viroporin, that there really should be more effort in understanding these proteins well. So viroporins are important as antiviral drug targets. They tend to be helical bundles, and they're oligomeric. The oligomer number is something interesting in itself, and it's not easy to determine. They're relatively small, and that is the main reason why they have not been well understood, because they're too small to crystallize. There's not enough water-soluble portions in these proteins for people to make nice crystals, and they're also really too small for cryo-electron microscopy. So these properties are what makes viroporins really terrific targets for solid-state NMR. Now, let me see if I can make this, okay. And so representative viroporins include the influenza M2 protein and HIV VPU, and now the SARS-CoV-2 E. As you know, these viruses have haunted humans for, many decades and centuries and have killed many people. So these proteins, understanding these protein structures are important both for fundamental reasons and for biomedical and public health reasons. So the SARS-CoV-2 virus encodes 4 structural proteins. Among these, 3 are membrane proteins. The spike protein, which is what makes these viruses have this very characteristic spike, crown shape, the membrane protein, and then the envelope protein. The E protein is the smallest of the 3. And if you look at the SARS-CoV-2 life cycle, you know how these 3 membrane proteins act and orchestrate their activities at different time points of the life cycle of the virus. So the spike protein is what causes the virus to, enter the cell. So spike S protein recognizes cell surface ACE2 receptors, which cause the virus to enter. And once inside, the viral RNA is replicated and transcribed, and new virus proteins synthesized, and the new viral RNAs and proteins are then assembled by the M protein at this compartment of the cell called ergic, and it stands for ER-Golgi intermediate compartment. And here, a new virus would bud into the ergic lumen, and that direction of budding is ensured by the E protein. And after the budding, the new virus would then get exocytosed out of the cell. So the E protein is important for proper budding of the virus, and that's its first function. But in addition, this E protein also conducts ions, cations, and it does so in a relatively non-selective fashion. Among the cations that it's known to conduct, calcium. Well, I should start with potassium and sodium, and then there's calcium, and this calcium conduction activity is interesting because there's literature's report that this calcium conduction triggers the inflammasome of the cell, which then causes all the symptoms of the SARS infection. And so hexamethylene amiloride, the small molecule drug, has weak inhibition against the E protein, but better drugs need to be made. So we would like to study this protein's structure and drug binding by NMR. So for those of you who haven't had to look at solid-state NMR or think about it, so solid-state NMR is a branch of NMR spectroscopy. And in NMR, what we do is to detect the oscillatory response of atomic nuclei that carry spins to resonant irradiation of radiofrequency pulses in a large magnetic field. And the frequency response of these atomic nuclei with spins shown at these various peaks in the chemical shift spectrum, they tell you the chemical structure of the molecule you're interested in. And through these chemical shifts, therefore, we get chemical structure information, whether you're looking at some kind of organic compounds or proteins or nucleic acids and so on. But in addition, to this chemical structure information, NMR spectra also encode three-dimensional structural information, and that's what I'm particularly interested in. And the reason that the 3D structural information is encoded in the spectrum is largely because the internuclear couplings, primarily dipolar couplings, depend on distance in this manner. So the dipolar coupling scales with the distance inverse to the cubic to the third power. And let me just dismiss this. So we can measure the dipolar coupling effect through intensities of the peaks that we see in the spectrum. Now, what is special about solid-state samples, in other words, samples that are not isotropic liquids that are tumbling in a tube, these hydrated solids, such as membrane proteins, they are not tumbling, the molecules are not tumbling fast. So in order to get really narrow lines, highly resolved spectra, we have to do something special. And that something special is the magic angle spinning. So we put our samples, a hydrated membrane proteoliposome, into a cylindrical rotor, which is tilted by 54.7 degrees, this magic angle, from the magnetic field, and we spin the sample around the rotor axis. And this is the way with which we get high-resolution spectra that look like or similar to the solution state spectra that you're familiar with in your organic chemistry class. Okay, so this is the basis of modern solid-state NMR spectroscopy. So what we do in order to study the SARS-CoV-2 E protein in the lipid membrane, in a membrane-bound state, in other words, we don't use detergents, we really use phospholipids, and moreover, we want to use phospholipids that mimic the cellular membrane in which SARS-CoV-2, a new virus, is assembled. And so in order to do this, first we purify the protein, and then we reconstitute the protein in a phospholipid mixture. And so Matt McKay and Shiva Mandela in the early days of the pandemic worked very hard to purify the E protein. And we focused on the transmembrane domain, which has about 30 residues shown in red here. And we used the SUMO tag to increase the solubility of this whole construct. And that's how we purified this protein in HPLC. And you can see in the gel that the SUMO tagged protein, as well as the subsequently cleaved ETM alone, the native protein, can be produced in a relatively pure manner. Now, for our spectroscopy, we also care a lot about incorporating fluorine, as I will explain later. And so here is has a mass spec that shows that we can incorporate fluorinated phenylalanine residues into the protein in the native phenylalanine positions. So what we do once we have these nicely carbon-13 and nitrogen-15 labeled E transmembrane domain is that we reconstitute the protein in lipid bilayers, and we look at the lipidomics literature to find out what kind of lipids are prevalent in the ergic or ER-Golgi kind of membrane. And you have your normal phosphatidylcholine, phosphatidylphenylmine. But in addition, we also have something somewhat special, and that is phosphatidylinositol. And then we have negatively charged POPS. And as true for most eukaryotic membranes, you have a certain amount of cholesterol. So we put in these lipids at the levels that are known for the ergic membrane. And our first study started with neutral pH. Okay, so, and you can see even in our very first measurement of the first fingerprint spectra, here you can look at this 2D nitrogen-carbon correlation spectra. You have very nicely resolved peaks. There are only 31 residues in this transmembrane peptide that we expressed, and we can already resolve in this simple 2D something like 2/3 of the residues. And we can assign these chemical shifts, these peaks, based on more extended three-dimensional correlation experiments that I won't show here. Now, the information that comes from these assigned chemical shift is the backbone conformation, in other words, phi-side torsion angles. And that's because there is a well-defined, well-known correlation between chemical shifts and the backbone conformation. And from these chemical shifts shown here as the deviation of the C-alpha carbon chemical shift from the random coil position, chemical shift, both C-alpha and C-beta, we can immediately tell with this trend that you have alpha helical conformation for most of the transmembrane domain that we have expressed. This is residue 8 to 38, so you have 31 residues, and this chemical shift trend indicates that we have a well-defined helical peptide, with the exception of the last few residues that this trend is starting to deviate from alpha helix. So the backbone conformation or the monomer conformation is relatively clear, alpha helical. But as for most oligomeric helical bundles, the critical question to sort out structurally is the quaternary structure. In other words, how do these helices pack together? What are the interhelical distances? And also, what is the helix orientation relative to the lipid membrane? And these two pieces of information are what is difficult to measure by NMR traditionally. And so I want to walk through the strategies that we have developed over the years in the lab to get these two pieces of information. And for SARS-E, we focus primarily on the interhelical distance extraction. And as you can see in these cartoons, our primary strategy is to mix carbon-13 labeled protein with a fluorinated protein at a 1-to-1 ratio. And these fluorines usually can be incorporated into aromatic residues in the protein relatively easily. And in SARS-E, we have only the transmembrane domain, we have only 3 phenylalanine residues. So there are only 3 fluorines that we can tag at the para position. Now, why do we care about fluorine? That's because fluorine has one of the highest magnetic dipole moments among all spin 1/2 nuclei. So here shown on the right are these circles that are drawn to scale to the size of the gyromagnetic ratio, which is basically about the magnetic dipole moment. And the higher the magnetic dipole moment, the stronger the dipolar coupling. If you look at the expression for the dipolar coupling, there's these gammas, which are the gyromagnetic ratios. The size of the coupling depends on the 2 nuclear spins gyromagnetic ratio. So if you can use a high gamma spin, you can measure the coupling to longer distances compared to if you use low gamma spins. And the traditional carbon-13 and nitrogen-15 NMR, that is the mainstay of NMR spectroscopy, just turns out to have low gamma. And so the distance measurement goes out to about 5 angstrom, and then any further is a real struggle. So fluorine overcomes that problem. In addition to this high gamma, fluorine is also naturally 100% there, fluorine 19, it's there all the time. So you don't have to enrich it compared to C13, which is only present at 1.1% in nature. And in order to do carbon-13 NMR, you have to enrich it. And so fluorine comes for free in a sense. And also it turns out that fluorine chemical shift is sensitive to the chemical environment, the chemical structure. And so that makes the frequency position very variable. And that's another advantage. Now, you know that fluorine doesn't occur naturally in proteins, and that is good actually, because that means we can specifically introduce fluorine at places we want, and you don't have too many of these, and you don't have to worry about assignment. You know exactly where you incorporate fluorine. So for all these reasons, fluorine NMR is really a very nice tool. Now, let me just focus on the measurement of long distances. We care to go to 1 to 2 nanometer range because if you think about oligomeric helical bundles, and that's the kind of range you would like to be able to measure. While if you only can see to 5 angstrom, you're going to be struggling to get any interhelical contacts. Now, fluorine-based distance measurements have been done before, particularly by Jake Schaefer's lab already started in the 1990s, but the methodology or the approach was relatively low throughput. And it's because the experiment was done in a one-dimensional sense, and you cannot resolve too many peaks in 1D. It's also done in the old technology at relatively low magnetic fields. And so you basically, the resolution gets worse and sensitivity gets worse at low field. So we would like to be able to do fluorine-based distance measurements at high magnetic fields, where you can resolve better, see more peaks, and where you also get higher sensitivity. So our basic design in terms of new advancing the methodology is to put these distance measurements in a two-dimensional template. So here in this example, we have a 2D carbon-carbon correlation experiment. And then within that experiment, we add a fluorine distance measurement module and measuring specifically carbon-13 to fluorine distances using these orange pulses. These pulses, fluorine pulses, come out in a rotation synchronized manner because we're meanwhile doing magic angle spinning. And these fluorine pulses are put out in order to recouple the dipolar coupling. So the bottom line is that we're going to measure two 2D spectrum. In one spectrum, we don't apply these fluorine pulses. That's our control, S0. And in the second spectrum, we're going to apply the fluorine pulses, and only those carbon-13 spins that are close to the fluorine, within about 10 angstrom or so, will have intensity change, intensity reduction. And that's our S spectrum. So by subtracting the two 2D spectrum, then we can see which carbon-13 sites are really near the fluorine. And that is basically our general approach. Now, in order to do this nice 2D experiment with a lot of site resolution, you want to go to high field, and high field in this case is a 600 megahertz NMR at 14 Tesla. And when you are at that field strength, we find that the fluorine chemical shift effect is quite large. So what we have to do to overcome that is to spin our sample fast. So here are simulations that show you that if you spin slow at about 12 kilohertz or so, then these dipolar coupling dependent distance curves, Redor curves, that technique is called Redor, depend on the depend on the size of the CSA, and so this is not so good. While if you spin fast, then it doesn't matter what's the size of the chemical shift for the fluorine, you can always get more or less near universal curves where the distance decay depend purely on, well, the Redor decay purely depends on the distance, which is what you really care. Okay, so we first demonstrated this 2D carbon-carbon resolved carbon-fluorine experiment on the model protein GB1, where we incorporated 3 fluorines into this protein, 56 residue protein. There are 3 tyrosines, and at each we basically added a 3F fluorine at the 3 position or meta position. And all you do is to incorporate fluorinate tyrosine in your protein expression in E. coli. And then you use glyphosate to suspend the pentose phosphate pathway of the natural sort of aromatic amino acid synthesis by E. coli, and you use the supplemented tyrosine for your incorporation into the protein. All right, so these are the 3 fluorine signals of the tyrosine, but the key experiment here is shown here. We have 2 2D spectra, carbon-carbon correlation, and this is the black is the control without the fluorine pulses on. And then here in red, I have already subtracted the S0 and an S, and I'm showing you only the difference where these signals come from those carbons that are nearby to one of the 3 fluorinated tyrosines. And so with this, we can now really quantify distances, I can measure these different spectrum as a function of how long I apply the read-or pulses, how long the train is. And so here Alex can simulate the intensity. So here's the S over 0, the D phase over control, and just 2 points. And you can simulate with these so-called universal curves to figure out the individual distances for these various spots that you can resolve. And a second long distance ruler that we developed that's even better than a carbon fluorine is to combine proton and fluorine. Proton and fluorine are the two highest gamma nuclei among all spin 1/2 stable nuclei. And by saying stable, I'm excluding tritium, which is a radioactive nucleus that you don't want to touch. And so tritium happens to have a relatively high gamma. But anyway, so if you look at the gamma of proton and fluorine, you could predict theoretically that one should be able to go to about 1.5 nanometer. And so here, the experiment that we designed is a 2D nitrogen proton correlation experiment. And you can see here we are detecting protons, so that's good. We can get higher sensitivity by detecting this very high gyromagnetic ratio spin. And then in this experiment, I again incorporate fluorine and proton dipolar recoupling pulses so I can measure the proton fluorine distances. So here you can see, again, we demonstrate on GB1, but the effect now is really a lot stronger in a sense. We have these nitrogen proton 2D spectrum that can be measured relatively quickly because of proton detection. These are done at 40 kilohertz spinning. Each 2D spectrum takes about 3 hours, and we can assign these. And then again, we turn on the fluorine pulses. We see some of these peaks will have lower intensity, and then we subtract. And here in this different spectrum with a short fluorine pulse train of 1.8 millisecond, only a few amide proton signals show up, and these are the amide hydrogens that are closest to the fluorinated residue. And in this case, the fluorine is put onto the single tryptophan, the only tryptophan in the GB1. And then if you can want to turn on the, or keep the pulse train going for longer, then more amide hydrogen signals will show up in a different spectrum. So more and more remote hydrogens will show up in the spectrum. So now if you look at the dephasing curves, S over 0, you can map out for different residues these different speed of dephasing that really correspond or tell you the distance. The shortest distances will have the fastest dephasing, the long distance will have slower dephasing. So in about 2 days, you can measure a string of these pairs of 2D spectra and can get all of these very quantitative distances, which is very powerful. So now we have a fast, long, and precise distance ruler. So we would like to apply this method to our membrane proteins. So now coming back to the SARS-CoV-2 E protein story, after the monomer, the backbone conformation has been clarified from the chemical shift assignment, Shiva and Matt made this such a sample of C13 fluorine mixed sample. And there are 3 native phenylalanines in the transmembrane domain. So each of them got a parafluoral label. So we have 3 labels in the spectra in a sample. And so in this case, we did the 2D carbon-carbon resolved CF read-or distance measurement. So I show here the control spectrum where all the carbon signals show up. And here's the different spectrum, and you can see which carbons in the next molecule are close to the fluorinated phenylalanines. We can also do 1D experiment to have more different mixing times. So this requires more sensitivity. So we did the 1D fashion here. To supplement these quantitative distance measurements, we did 2 more experiments to figure out the topology of the helical bundle. And that topology means, in this case, which residues face the water-filled pore versus facing the lipids. And if you think about it, in any helical bundles that can conduct ions, there should be some water molecules inside. And so what we do here is to transfer the water proton magnetization to the protein in this way, and we detect the result of that transfer through protein spectrum. So any water-accessible pore-facing residues should have higher intensities, and that's what we're showing here in this sort of a color-coded spectrum, 2D spectrum. Shown in blue are the higher intensity water-transferred residues. That means these residues, side chains, are preferentially facing the pore, and shown in red or lower intensity are those that are dry, therefore facing lipids. And we can flip this experiment to select the myelinization from lipid protons and then transfer those to the protein. And when you do that, then you are picking up those residues that are lipid facing. And here, if you look at this 1D spectrum that came from such a lipid transferred myelinization transfer, then these phenylalanine side chains look very strong in intensity. So they're preferentially lipid facing. So when you combine all of these interhelical distances from fluorine reador and these pore facing versus lipid facing topology measurements, as well as the backbone conformation from chemical shifts, then you can calculate the structure of the E transmembrane domain using Explore NIH. And so here is the helical bundle. It's a 5 helix bundle, and I will explain why it's 5. And you can see it's a relatively tight helical bundle. The pore diameter across is about 10 to 12 angstrom or so. And the helices don't have a particularly well-defined handedness. It's not a clearly left-handed or right-handed. There's some disorder along the way, and its overall look is close to vertical. The pore-facing residues are highly hydrophobic, or methyl-rich sort of ILV residues, except for the N-terminal. There's a few polar residues, including this glutamic acid. And also including a asparagine 15, which is also in the N-terminal segment, and it looks like it's facing the pore. So this may be important for ion conduction. And in terms of these phenylalanine residues, they're all in the middle of the transmembrane segment, and they seem to stack across the helix-helix interface. So this pi stacking is interesting. And we subsequently did experiments to show that the aromatic side chain conformation actually changes between the closed and open state. So these rings can move or reorient somewhat to perhaps loosen or tighten the helical bundle to regulate the ion conduction. Okay, so now coming to the question of how do we know this is a 5 helix bundle? This came from the literature for about, I think, 15 years before the COVID-19 pandemic. A group, or two groups primarily from Singapore and Spain, they studied the SARS-CoV-1 version of E protein, which is highly homologous to SARS-2. And so various attempts to use SDS page gels or gel electrophoresis to find out the oligomer states have been made. But these are just a few examples, and there's also analytical ultracentrifugation data. But as you can see, the gel band really is not that clear. And also these studies changed the detergents quite a lot. And they try to count the number of bands, and they think the maximum is around 5. But these data were not convincing enough to the field. So we get asked questions, are you sure this is really a pentamer? And so this is actually a general problem for viral porins or virus membrane proteins. When they're small, the stoichiometry of assembly is actually pretty difficult to measure. And so it turns out that my group actually has thought about this problem for quite a while before the pandemic, where we developed a strategy to measure the oligomeric state of membrane proteins directly in lipid bilayers. And again, the go-to nucleus is fluorine. And here the idea is the so-called fluorine codex experiment. And what we're doing here is we are measuring a spin echo after we transfer the fluorine magnetization from one spin to another. And if this magnetization transfer is happening, usually in a non-crystalline sense, then the chemical shift orientation will change between the two. And this will, because of the design of the spin echo experiment, this means that the echo intensity will drop if there is this magnetization or chemical shift tensor orientation will change. So if you have, let's say, a trimer, at the end of that magnetization transfer process, equilibrating the magnetization among 3 spins, then the initial unit or the initial magnetization that remains on the source spin should be 1/3. That's the portion of this magnetization that doesn't change its orientation. And then my spin echo experiment should have a 1/3 of the intensity compared to the original situation. So if I have a tetramer, then the remaining magnetization or spin echo intensity should be 1/4. And if I have a pentamer, then I should have 1/5. So that's the design of this so-called codex experiment. So if SARS-E indeed forms a pentamer, I should expect 1/5 of the spin echo intensity once my magnetization is well equilibrated and really has transferred. And so Noah in my lab conducted this experiment on ETM. And here we took more time to make sure we only have really just one fluorine in each peptide. And we put this fluorine at one of the 3 phenylalanines. So we did this Codex experiment. You can see the echo intensity drops and equilibrates. And we initially stopped at about 2 seconds. We're happy to see 0.2. And then we thought the intensity looked still quite good, so we went further. And then by the time we got to 4 seconds, we were surprised to see that the intensity actually dropped below 0.2 to 0.14. So if you do the algebra, you say, oh, wow, this could be a heptamer, 7 spins. And so that was surprising. And is that really 7 spins? So then we thought maybe we have too much protein in our membrane, so 1 to 17 mole ratio. And perhaps you have clustering of channels that will confound the matter. We care about the subunit oligomer number, not having multiple channels glued to each other. So we diluted the protein by a factor 2, and now you see this is 0.2. So this is more clear to be a pentamer. Nevertheless, the fact that at 1 to 17, the 1 to 17 mole ratio goes to 0.14 clearly says that you can have multiple pentamers being very close to each other. And so that is a clustering phenomenon, which is interesting. And remember that fluorine distance ruler cannot go beyond 2 nanometer. More than that, it's very hard to see. So 2 pentamers being together is still quite close within 2 nanometers. And so we thought this clustering is interesting. What is causing it? So we started changing the membrane composition. And by the time we remove both phosphatidylinositol and cholesterol, we find that even with the same protein concentration, protein-lipid ratio, the lowest equilibrium value is actually 0.2. So we don't have that phenomenon of 7 spin in close proximity. So this is a thread that we're following up more, that these pentamers can cluster in a phosphatidylinositol containing membrane, and this could be interesting or maybe relevant for the function of these E proteins. So this is some Monte Carlo simulations about, what kind of distributions one might have in a sort of a given proper or reasonable force fields, and how these pentamers might cluster in a membrane. And each of these blue dot represents, let's see, each blue dot is actually a pentamer. All right, so okay, so then Joao in my group decided that we're interested in understanding how this channel actually opens. And so if you look at the ergic lumens chemical composition, you find that it's acidic and it's rich in calcium compared to the cytoplasm. And so proton and calcium would be the 2 ions that may play a role. And so we showed by direct NMR experiments that if we now change our sample, set it up at low pH or neutral pH, but adding calcium, or having low pH and calcium, then the water transfer to magnetization, the water transfer happens a lot faster. So what we're plotting here is the intensity of the water transferred protein signals as a function of the transfer time. And you can see that both ions, or each ion, does a better job of filling the channel pore with water compared to the neutral pH, no calcium situation. So this becomes then the stimulus to open the channel. And indeed, if you now prepare a sample, or measure a spectrum of E under this acidic condition in the presence of about 20 millimolar calcium, then this on the right is the 2D carbon-carbon correlation spectra, where the intensity is fully transferred from water, and shown the more blue and green you have, the higher the intensity, while the neutral pH sample without the calcium, the water transferred magnetization, the intensity is low, as shown by the red nature here. So the color coding is telling you how high the peaks are. And so, so think of it this way, that a neutral pH, we have a closed channel with a narrow pore without too much water in there, and a low pH and in the presence of some calcium, you should have a wider pore with more water. And so now Joao took all of these fluorine distance measurement tools, whether it's carbon fluorine or proton fluorine, and made the sample to do these interhelical contact measurements. And again, to reiterate, the proton fluorine distance we're measuring is between the Mi proton of one helix to the fluorine of the next helix. And carbon fluorine data is shown here. But in addition, he also supplemented these fluorine-based distances with traditional carbon-nitrogen-based sort of interhelical contacts by playing the game of mixed labeled samples. Examples are shown here. All right, so now another one new piece of information or really powerful constraint and that we measured on these low pH samples is that now we obtained helix orientations. As you can imagine, that these helical bundles, if you can figure out which way, how tilted these helices are, that is a major piece of constraint. And so the way we measured this is to ensure or put this protein in a relatively low viscosity membrane, POPC, POPG, in which the helical bundle is doing whole body uniaxial rotational diffusion around the membrane normal because this lipid mixture is very fluid. And under that rotational diffusion condition, if you measure NH dipolar couplings under that motional averaging situation, then what you find is that the NH dipolar coupling will reflect the helix tilt around the bilayer normal. So that data is shown manifested here. This as a function of the residue, we're basically figuring out the NH bond orientation. And for most or 2/3 of the residues, it's about 5-degree tilt, and then at the end, it's tilted slightly more. So then now you can combine these extremely powerful orientational restraints with the interhelical contact measurements, both quantitative and semi-quantitative. And now here's the high resolution structure of the open state. And you can see how this 5-helix bundle now has a pretty different shape. The N-terminal 2/3 looks like a wide helical barrel instead of a narrow pore. And then the C-terminal 1/3 kind of pinches a bit close or shut in a sense. And that compares with this neutral pH sample where it's kind of throughout relatively narrow. And if you look at the water-permeated pore, the pore profile is kind of reversed between the low pH sample and the neutral pH sample. The low pH sample has a wide pore at the top, and then there's a position here that's kind of tight. And that is probably the most hydrophobic, we think, the gate of this channel. Okay, so what kind of mechanistic insight can we get about the ion conduction mechanism now that we know the structure of the open state at low pH? So it turns out that there are really two polar networks in this transmembrane domain. The N-terminal, about 7 residues, starting from a glutamic acid to an asparagine, there's a serine. There's just a bunch of very much, hydrogen bonding groups in the side chains, and then there's this titratable. That's the only titratable residue in the entire protein. And if you look at the lowest energy structure ensemble, there seems to be 2 types of N-terminal entrance configuration. In one configuration, this E8 side chain, this carboxyl group, reaches out to the next helix threonine. And so you can imagine if there's water or calcium coming in to break this, that could cause ion conduction. And then in the other configuration, the second form, this E8 side chain carboxyl reaches out to the top to receive potentially protons to be protonated. So that's information that's interesting. And so we think that proton and calcium ions could be recruited and relayed through this hydrogen bonded network down the channel. Now, then there's a vast segment of about 20 angstrom or so that is all hydrophobic, and we still don't fully understand how ions are conducted that way. But we can at least say that when you reach near the C-terminal end, there's an arginine residue. And this is interesting, that in the open state, so-called open activated state, this arginine side chain guanidinium group is swung into the channel pore and reaching out to the next helix of threonine. So this part of the channel becomes extremely polar and hydrophilic, and we think that could help to reduce the potential mean force or energy barrier for passing ions, cations, past the most hydrophobic sort of position of the channel because just how polar this region is. And if you look at our closed state structure solved earlier in 2020, that arginine side chain is actually flipped out to the lipid-facing side, interacting with the lipid phosphate through favorable salt-bridge interactions and so on. So this is a very interesting change, and we now have more recent data that shows indeed that the open state or the low pH sample, the arginine side chain is slightly more rigid, while the neutral pH sample, the arginine side chain is more dynamic. So more recent work involves actually looking at a T9I mutant of the E. So now, again, remember the T9 is the second residue in our sample. And in the Omicron variant of SARS-CoV-2, this T9 is predominantly switched to an isoleucine. And biochemical data from this paper, Shah et al., shows that this T9I E protein is not as good of a ion channel compared to the wild type. If you switch to low pH, this chart shows that it doesn't conduct as well as the wild type, which is more acid activated. And what is interesting when we looked at this closely is that if you look at the T9I 2D nitrogen carbon correlation spectrum, so the shown in black is the wild type. And I need to emphasize that these spectrum is measured for solid, emphasizing rigid residues, immobilized residues. So if you have high intensity, that means the sample or the residue is rigid. So T9 or the wild type in the neutral state, which is non-conducting, we have strong signals for all the residues, particularly, for example, T11. And we don't see the T9I's red sample signal here. When we switch to the low pH sample, which is the open state, the wild type protein doesn't have the T11 signal, and the wild type is now actually a functional ion channel, but the signal is gone for T11. While the T9I mutant, the T11 signal is there, indicating that this residue is rigid in a mutant. Now, remember, the mutant doesn't conduct ions very well. So what this means is that dynamics supports function, and this mutation rigidifies or immobilizes the N-terminal segment somewhat, and that's correlated with lack or weak activity. So I'm going to skip some of these and just emphasize that what we learned is that there's a whole set of these polar residues in the N-terminal region which interconverts kind of between different configurations or chemical structures in a very dynamic way to relay and recruit and relay protons and calcium ions. And the interactions among these polar residues are sensitive to, whether you have the threonines around, this T9 particularly. And it's also sensitive to the lipid environment. So I'm not showing you the data, but we have looked at model membranes without all the ergic component versus the complex membrane with cholesterol, with PI, and so on and so forth. And the chemical shifts are different. And so the polar networks ion conduction depends on the environment significantly. So now in the last part, I want to say something about our studies of the drug binding to this E protein. So now that we know the structure for two states, the closed and open state, the interest, of course, is, okay, what kind of drugs can one design to block this channel or inhibit, inhibit this channel? And coming back to this hexamethylene amiloride, so you have this amiloride moiety here, it's a hexamethylene group. Although it's a weak inhibitor, it is the starting point for us to figure out the binding mode to the E protein. And so we have collaborators that produce these nicely C13M15 labeled drug for the polar end of the molecule, and also give us nicely fluorinated drug for the hydrophobic end from different groups. And as you can now have a sense of with NMR, if we can label different components in an orthogonal way, it's really advantageous. And so here, what we have done is to label protein, for example, C13 and drug N15, or protein fluorine and drug carbon, or vice versa. And so I will show you some data that allows us to figure out where that hexamethylene amiloride HMA is actually bound to this E pentamer channel. Now, I want to prepare you, though, a little bit. When we look at these small molecule hydrophobic or amphipathic small molecule binding to these membrane proteins, often it's actually quite difficult to figure out the binding stoichiometry. And that is if you titrate in drug, let's say one drug for each protein, which means you have 5 equivalent of drug to this pentamer, to the pentameric channel, exactly whether all 5 will bind to a channel or maybe one and so on. That is something that's not easy to tell actually from any. There's no simple biochemical method to get this information in the whole membrane lipid bilayer. So what we do is to use ReDOR. Again, we come to this very nice technique. And so you can imagine if you observe the drug spectrum and you run your ReDOR experiment, meaning your protein is dephasing the drug. If in this 5-to-5 situation, all 5 drug is near the protein, then the protein will dephase all your drug signal. You should expect a curve like that. But if only one drug is within the proximity of the protein, then only 1/5 of the drug signal should actually see an impact. And so from the drug detected ReDOR spectrum, you can figure out right away what is the binding stoichiometry, all right? And now if you suppose you have a stoichiometry of one drug per channel, but then you have the question, is that drug centrally located right in the pore shared by all 5 helices, or if the drug is actually peripherally bound perhaps to one of the helices and not far away from the other, how do you tell that piece? And to do that, we detect the protein signals, protein detected ReDOR, and if you have a central drug, then all your 5 helices should be affected by that one molecule of drug. So you should expect a blue curve. But if you only have one protein bound to the drug, in a sense, drug in the peripherally bound, then you would have this magenta situation. So by looking at the drug and the protein spectra one by one, you can figure out both the binding stoichiometry as well as the chief mode of binding. And so here's the first piece of data. In this case, we have a fluorinated protein combined with this C13 and N15 labeled drug, and we're detecting the drug signal, ReDOR. And we have 2 samples. In one, we have 5-to-2. So in other words, 1 pentamer to 2 equivalent of drug. In the other sample, it's 5-to-5. And here's the difference signal indicating, yes, we indeed have a drug in contact with the protein. But the interesting piece is here, if you have the protein in this 5-to-2 sample, my drug signal decays to only about 0.5. This is a simulation curve. In other words, only one of the 2 drugs is actually impacted by the protein. And that means the stoichiometry is 1 drug per pentamer. If I have 5 drugs into 5 protein in my sample, then my drug signal decays only to about 80%. So that again is saying 1 drug per pentamer. And now I switch to the protein detection, and now we're observing protein signals, proton, nitrogen, and so on, and I have a fluorinated drug, and we're looking at the decay of the protein signals by the fluorinated drug. Now you see how high these intensities hang, the decay to about 0.9, 0.8-ish, which means I'm not having a drug in the middle of the pore, and rather it is a drug which is peripherally bound to the protein, to one of the helices. And so when these are the key qualitative motifs and the rest are details, we can look at which residues are close to the fluorinated drug. And here's basically docking under the constraints of all the NMR observed contacts in a semi-quantitative way. And we have different poses coming out of the docking, and you can see the so-called bridging mode. Here's the top view. You have the drug slicing. It's down the membrane with the plane sort of parallel to the bilayer normal. And here, in this case, the hydrophobic hexamethylene ring is pointing up, and the guanidinium polar end is pointing down, closer to that arginine at the downside. And so here are some of the favorable contacts. And then you have what I call parallel pose, where the plane of the drug molecule is really kind of parallel to one of the helices. So that's an interesting pose. And then here's what I call tangential pose, where the plane of the molecule, the drug, is now parallel to sort of two helices, and there's a way of intercalation. All right, so but in all of these poses, slightly different poses, it looks like the drug is inhibiting this channel in a sort of allosteric way. It doesn't really go inside the pore. It certainly doesn't. Our data shows that very clearly. We think that this is the inhibition mode is by tightening the channel. So most of these data were actually collected at low pH corresponding to the open state. So we think that the channel would be just sort of tightened by the HMA sitting in this way. So this is the summary. So I hope I have shown you that with modern solid state NMR, particularly these very effective long distance probes, including fluorine, that we can not only solve oligomeric helical bundle structures for these virus porins, which really cannot be touched by alternative structural biology techniques, but we can also figure out drug binding. So with that, I want to thank Joao, Shiva, Alex, Matt, and Noah for all these data. Yeah. So, and I would like to thank you for your attention.
Thanks, May, for the excellent seminar. Now I'd like to just open up the floor for any questions anyone had for May. So please just turn on your cameras or your microphones if you want to ask, or write them in the chat box. I can start off though while we're just waiting. I'm really interested in how the composition of the phospholipids in the membrane can influence the conformation of the helix bundle. In particular, you said in your codex data that it may influence channel clustering. Is that something you're investigating more, or how do you think that might influence the function?
Yes, we are investigating more. We're very much chasing that thread. It's very interesting to us. Phosphatidylinositol is negatively charged. It has an interesting sugar ring, which might interact with the glutamic acid in some way. And PI is something that we never had to use before when we studied influenza M2, which buds from the plasma membrane. There's almost no PI in the plasma membrane, but in ergic you have it. And PI in its phosphorylated forms, as you know, the inositides is a signaling lipid, and signaling, and so this could have biological consequences. You know, if E channels, these viroporins are clustered together where PI is enriched, maybe this would have something to do with the triggering the inflammasome. Yeah, so we're guessing. We're using NMR to figure this out. This is a very nice problem for us. Yeah, it's a good scientific question for us. Yeah.
Thank you. Isabel, your question?
Beautiful talk, Mayan. Really, really nice. Guess I have a couple of questions, maybe more practical. How much protein do you need for solid state NMR?
Yeah.
That would be one question. And the other question is, to incorporate the fluoride, do you use a natural amino acid? Or can you get the same result if you just crossing the fluoride?
Right. Let me tell you. So for ETM, which is really a peptide, a small peptide, we can get away with about 2 milligram and still measure a lot of spectra. The highest we would go is probably 5 milligram. It's not fully necessary. And in terms of now, for bigger proteins, if you go to something like, let's say, full-length E, okay, there I think we can still do the spectroscopy with probably 5 milligram. And I can tell you that with our expression and purification protocol using SUMO, tag, and then cleave per liter of M9 minimal media, we can make anywhere between 10 milligram of the short construct ETM, and sometimes even more. Okay. Fluorination in my group, we have so far only done the very simple method of glyphosate to basically replace the natural pentose phosphate pathway production with our fluorinated aromatic amino acid. And so you have to add all three aromatics, tyrosine, phenylalanine, and tryptophan, and all of them would be natural abundance in terms of carbon-13, but usually we would say, okay, let's add a parafluorophenylalanine or 5-fluorotryptophan is our favorite. Tyrosine we don't usually do because it's a bit more chemically sensitive. You have an OH group I don't want to mess with. But the unnatural amino acid is a strategy that other groups have done, and we're now starting to collaborate with people who are doing this. Yes. And that becomes relevant if you start to have, a big protein with so many aromatic residues that you cannot afford to label, or you don't want to label all of them because that might start to cause perturbation to the protein structure. In general, we don't want to incorporate too many fluorines in the protein. And so, yeah, we haven't had to do this, but our collaborators are happy to do this. Yeah.
Thanks, Nate.
Yeah,
Tracy,
please.
did you?
Yep, hi mate. That's an amazing story. It's really great. I was just wondering, does this follow on that fluorination pathway? Do you have any sense if adding the fluorines changes the pentamer in any way, or
if what changes?
By adding the fluorine, does it change the pentamer, how it interacts, and things like that?
Oh, okay. Whether the pentamer is affected by the no, we know it hasn't affected, and the reason is we have enough samples that we don't. Well, our regular 2D carbon nitrogen correlation spectrum, we measure that without fluorine, right? So we know the chemical shifts. And then if we say, oh, let's make a mixed label sample, well, guess what? We can still measure those carbon nitrogen spectrum. So we keep our eyes open about whether there's any chemical shift perturbation. And if we don't see perturbation, then we say, okay, that's fine. So you have to be careful. And that's why in the first study for the closed state, where we try to get our result fast, we didn't have time to wait to synthesize the peptide with just one fluorine. We later took, when we did all these codex measurements, we synthesized that 31 residue peptide in order to not cause too much issues. Yeah. So I think right. So if you have a bigger membrane protein, then our simple-minded synthesis obviously wouldn't work. And you really start to have to think about mutations or, things like that. If you can get away mutating away, except for the one you want, then that's also fine. Otherwise, it's unnatural amino acid. Yeah.
Thank you.
Yeah. So I think, though, coming back, I find the fluorine strategy to be the best in looking at ligand protein binding problems. That's where 30% of small molecule medicinal compounds already come with fluorine anyway. People have already done that change. And so I don't have to worry about causing potential issues with my protein. Yeah. Or doing a natural amino acid, which is still quite some work. Yeah.
Cool. Thank you.
Thanks. We're kind of running out of time, but there's one more question in the chat, a technical question, which says, thanks for the interesting talk. Can I ask what kind of solid state NMR rotor you use for 5 mg of protein?
Yeah, good question. So for regular carbon nitrogen kind of detected NMR, we use 3.2 millimeter rotors spinning at 10 to 15 or 10 to 14 kilohertz. For the fluorine, anything involving fluorine, we have one workhorse probe at 600 megahertz, and that's a 1.9 millimeter rotor that takes about 10 milligrams stuff. And that one we spin anywhere between 20 kilohertz and as fast as 40 kilohertz. And that's good enough for most purposes. Yeah.
Thank you again for a really interesting talk. Unfortunately, that's all the time we have today. And thank you everyone for coming to listen, and hopefully we'll see you all again next year when we start back up again.
Okay, thank you.